Hi everyone. So this is chapter 14, okay, molecular genetics. There are 4 parts to this chapter which I break into. I will only be covering part 1 in this video recording, okay? And this part is the part 1 and part 2 are actually quite abstract. So I don't want to talk about part 2. I just want to talk about part 1 for today, okay, or for this session. So let's start with this part on chromosomes, chromatin, and DNA. There are 3 learning outcomes that you need to know. Okay. Now the first one, right, will be a little bit hazy to you, but as we go on with the other chapters on molecular genetics, you will get to understand the terms better. And right now, if it's a little bit too abstract, it is okay. Okay, so don't worry so much. The main bulk of this session, right, is on the structure of the DNA, as you can see in outcome number 2 and number 3. Okay, so let me do this. Okay, so these two, right, will be on the structure of the DNA, and the first one, like I said, probably will be better as we go along. So what is found in the chromosome? Now, I think many of you, right, know what chromosomes are, okay? And some of you also know the term known as chromatin. Now, chromosomes and chromatin, right, actually refers to the same thing. Chromatin, right, are actually structures that is found in the nuclei of animal and plant cells, all right? And chromatin, right, actually is DNA, okay? Because it contains the genetic information in a molecular form, and this molecule, right, is known as DNA. Now, when do we call them chromosomes? When a cell, right, is undergoing cell division, the chromatin in the nucleus will start to condense and become very, very coiled, okay? And so this is known as chromosome. If you take a look at this picture, right, you can see the relationship of chromosome, chromatin, and DNA. And if you take a look at the picture, you realize that actually it's the same thing. Okay, it is just different terms used to describe different stages. So this is DNA, right? You can see it's very familiar to you. And then you have chromatin, which is a condensed form of DNA. And the chromatin, right, can condense even more to become what we call a chromosome. So how does a DNA molecule, right, undergo so many coiling and condensation, right, to form a very thick structure known as a chromosome? Okay, so this diagram on the right shows how DNA is slowly condensing to become a chromosome. You can see that the DNA over here, right, they are condensed around special proteins known as histones. Okay, histones. So this DNA, right, will coil around histones, and then these histones, right, will continue to coil until it reaches a second form of a coiling, and this is known as chromatin. The chromatin, right, will continue to coil and coil and coil or condense to form a very thick structure. Okay, and therefore this is called a chromosome. So actually, right, like what I said just now, they are actually talking about the same thing. Okay, now you can watch this video that I have sort of like sourced from the internet, and I'm not sure whether it works, but if you can see from the visual point of view, right, that DNA is actually undergoing a lot of coiling to form what we call a chromatin or chromosome. So what is DNA? Okay, now DNA is a legit term, so you can use DNA, but you also need to know the full name for it or the full term for it. And DNA is known as deoxyribonucleic acid. Okay, you must learn them. It's not for fun, so you must learn how to spell. Okay, so this is a molecule, and this molecule is the one that contains genetic information of the organism, and it is found in all of us. Now, a DNA structure, right, is a very familiar structure that you're looking at over here on this one. Yep, you can see this, right? And it's very familiar to most people in social media, in movies, so on and so forth. Okay, so how do you actually describe the DNA molecule? Now, actually, there are 2 strands of what we call polynucleotides. Okay, so you can see over here in this picture, right, the red color strand and the blue color strand. Okay, each of them, right, is known as a polynucleotide. And these 2 strands, right, are actually stuck together and they are twisted. So we call the overall structure, right, a double helix. You probably heard of double helix and DNA, and this is what it meant. Okay, so a double helix structure of a DNA, right, is actually made out of 2 strands. of what we call polynucleotides, and they twist around to form what I normally call it like a twisted ladder. These two strands, right, which you will learn later, are actually anti-parallel. Okay, now a DNA strand, right, a polynucleotide or one strand of a DNA is actually running in one specific direction. Okay, the reading of the sequence, right, is actually in one direction, and these two strands, right, are actually running in two directions, in opposite directions, and therefore we call them anti-parallel. So let's take a look at the structure of a DNA more. All right, now that this is the twisted form, right, and then now we are going to untwist it, and you're going to lay it out flat on the table like this. Okay, so you're looking at it right now. Okay, so you will have two strands of polynucleotide. All right, so let me mess up this diagram. Okay, yeah, this is one polynucleotide, and then this is the other polynucleotide. Okay, so it looks like from a molecular point of view, right, it looks a little bit weird, and yeah, so this is it. Okay, now the reason why it's called polynucleotide is because it's made up of many what we call nucleotides. Okay, so this nucleotide, right, consists of one sugar, which is what we call a deoxyribose molecule, okay, which is the one that is highlighted in pentagon with the letter S, one phosphate group, which is the circle with a P, okay, and one very unique nitrogen containing base, or what we call a nitrogen base. So these three, right, makes up what we call a nucleotide. And many nucleotides will form a line called a polynucleotide. Okay, so these are the terms, right, that will overwhelm you at first, but along the way, right, it shouldn't be a problem, I hope. Okay, and this is actually the basic, but never mind, let's try. All right. So this is known as a nucleotide. So let me ask you a question right now. Okay, now you take a look at this segment, right, or this thing, this DNA over here, right? Can you tell me how many nucleotides there are? Okay, can you get it? So there is one here, 3, 4, 5, 6, and then times 2, right? So altogether in this diagram, right, there are 12 nucleotides. Now the phosphate and the sugar, right, of one nucleotide, right, actually binds to the phosphate and the sugar of the next nucleotide. Okay, and this is known what we call a sugar phosphate backbone. You can see over here, right, that this is a nucleotide here, and this is another nucleotide, right? You can see, right, that this phosphate, right, is bind to the sugar of the next nucleotide in such a way, right, that it creates something like this, right, a zigzag kind of thingy, and this is the what we call the main structure, okay, and is what we call sugar phosphate backbone. Okay, so this is known as the sugar phosphate backbone. The difference, right, in the nucleotide, right, is not in the difference in phosphate and the difference in sugar. It's actually the difference in the what we call the nitrogenous base, the third component of a nucleotide. Okay, and this is the one that makes the nucleotide unique. There are 4 different types of bases. You probably heard of the term ATCG before. Okay, and this is the full term. A is known as adenine, T is known as thymine, C cytosine, and G is guanine. Okay, so because there are 4 different types of nitrogenous base or nitrogen base, nitrogen containing base, there are 4 different types of nucleotide. And actually, right, the whole DNA, right, is just made out of these 4 letters, which is amazing, and if you ask me, okay, because it's a bit ridiculous also, because me, you, and insect, a worm, right, we all contain the same 4 letters, but we are all so different. So let me ask you again, okay, like a little test. All right, this picture, right, how many nucleotides are you looking at? Okay, yes, actually, if you are well adversed right now, or you are well versed right now, right, it's 6. Okay, you're looking at the one half of or rather, you are looking at one polynucleotide with 6 nucleotides. Okay, now in order for the 2 polynucleotides, right, to stick together, to bind together, right, you can see that they are actually bounded together by hydrogen bonds. Okay, so over here, right, you can see that this is one nucleotide, and there is another nucleotide on the other side. And the reason why they are stuck together with a double strand, right, is because they are connected with what we call hydrogen bonds, okay? And this is known as a base pair. So a base pair, right, is two nucleotides that is opposite each other, and they are bound to each other by hydrogen bonds. Okay, now adenine, right, and thymine has a unique pairing, and they have two hydrogen bonds. Cytosine and guanine, right, if you can see over here, they have three hydrogen bonds. Okay, now this is consistent throughout. It is not through a stroke of randomness. So this is actually quite important in higher biology. Okay, so adenine and thymine has two hydrogen bonds for each base pair. Cytosine and guanine has three hydrogen bonds for each base pair. Now this bond, right, if you look at the picture, right, it's actually quite unique. It is consistent as well. Adenine, right, is always bound to thymine. Cytosine, right, is always bound to guanine. Okay, let me use letters lah. A is always bound to T. G is always bound to C. Correct? And vice versa. Okay, now we call this complementary base pairing rule. Okay, now this is the same for all, and if they are not bound in this way, right, the DNA will not work. Okay, the DNA will not be able to be consistent throughout, which we will talk about it in future under mutation. So adenine will always bind with thymine, and cytosine will always bind with guanine. Okay, so this is the last slide, and actually it is a very straightforward kind of a lesson which has no meaning at all, which is to talk about just the structure of a DNA. Okay, now if you do not understand, I perfectly get it, okay, because it's so abstract and it's like a molecular, it's like chemistry, right? So yeah, but never mind. Keep on doing this, and after a while, right, you realize that actually is okay already. Okay, but for right now, for the past 12 minutes, right, you will find that it is actually very abstract and a little bit dry. Yeah. Okay, so yeah, that's it. Okay, for this recording, and I hope you understand a little bit of this and it can help you in your March holiday assignment. Okay, thank you. Bye bye.